All right, so thank you for the introduction. Thank you, Derek, for the invitation and this wonderful event. And I would speak mainly about applications in neuroscience, since it became very frequent in neuroscience to use diffusion tensor imaging as the method of choice in order, alongside functional MRI, in order to see relations with behavior and cognitive experience and stuff like that. And the title of the talk is going beyond the diffusion tensor, because it's high time that we'll do because of many things that I will speak on. But just to start with, I would say the diffusion tensor imaging is great. It made huge advances in the way we can look at the microstructure of the brain and relate it to different cognitive experiences and things that are studied in neurobiology and neuroscience. And just to mention a few, with diffusion tensor imaging, it was revealed that at least the white matter is as dynamic as any other component in the brain, something that was not well understood before we were able to image white matter as we do with diffusion tensor imaging. It enabled, for the first time, in vivo quantification of the connectome. The connectome, the vast majority of all connections that we have of the brain. Obviously, it goes in different scales, but it allows, for the first time, the in vivo characterization of that. It provides characterization of pathological states in neurodegenerative diseases. Some of them we could not find with other techniques with MRI. So obviously, all these things made a huge advances with diffusion imaging. And we could at least say that DTI puts in vivo brain microstructural characterization at the front line of neuroscience alongside with other methods that are used. Before I go beyond the tensor, I just want to give three examples on that line, how DTI changed things in neuroscience, at least from my perspective, and to see where the beyond the tensor can help us get even more information. So the first example I want to show is about the dynamic brain. So it was shown by us and by many other groups that following cognitive experience, mean diffusivity reduces, not just only at the time of the activity, as Denise said, showed yesterday, but also following that. So you can measure hours, days, and weeks after cognitive experience, and in the relevant brain areas, the mean diffusivity will still be reduced compared to other areas. So that was shown both in animals and in humans in several studies. But it's not only that in the gray matter where you have the activity, things changes. And what things changes, we'll see in a minute. So something happens there that causes structural changes that reduces, alters the diffusion properties. But not only in that, since diffusion imaging gave us now a window to look at the white matter, several groups, that this is example from the groups from Oxford, showed that the fractional anisotropy does change following some kind of cognitive training that was in juggling, and we have shown in other cognitive domain of the memory that you do see traces in the mean diffusivity, in the FA, within the white matter, indicative of a new process in neurobiology, probably a process that appears in the brain, but finally can reveal it, of white matter plasticity. So this is the first example. The second example is about the connectome. So the connectome is just defining the connections that we have in the brain, and there are several frameworks that was led by Hagman and Spons about how to extract the connectome. So the connectome spans for different scale. It can be at the scale of a neuron, how many connections that it have, etc. It could be in the level of a column, of a region, or the entire brain. And diffusion tensor imaging combined with tractography allows us to look at that and try to get different information about how the brain connects at different people, at different scales, and in the work that we have done at different animals. So we have scanned several animals. Let's look what are the characteristics of the connectome that are well preserved through evolution, and just to compare the different brains of the animal. And indeed, the connectome is a very, very strong property of the brain in terms of graph theory. If you look at the efficiency of the connectivity that we have with the brain, is very well preserved through evolution. So obviously, this is the first glance of information that we can get into these properties, again, with diffusion imaging. And the last example that I want to show is in neurodegeneration. So we saw yesterday that the quantum leap that was in understanding stroke with diffusion imaging in animals and in animal models and then in humans, which made a huge benefit to society, but also in other diseases that are less understood than stroke, such as in multiple sclerosis. Using diffusion imaging, we were able to show, and many others were able to show that the extent of damage in the brain or the abnormality in the brain is far beyond the level of the lesions that we see in regular imaging. And that term coined the term normal appearing white matter that appears to be normal but obviously is abnormal, and extended, for example, in multiple sclerosis, the definition of the disease to be a disease not of lesion but a disease of the brain, the entire brain, either if you have lesions or not. So obviously these three examples show how diffusion imaging and tensor imaging allowed us to get much more information in neuroscience and in the neurology and define new concepts in the field. So please remember these three examples because we will come back to them through the presentation with how the beyond the tensor help us to understand other things, but why we need to do something beyond the tensor once we have these wonderful examples the diffusion tensor imaging was able to achieve. And the thing is that there are some limitations to diffusion tensor imaging that we need to be aware of. So the first thing is that it fails to represent the underlying microstructure in heterogeneous white matter. So we all know about the problems when we have one fiber orientation within the voxel, where you have crossing fibers, kissing fibers, dispersing fibers, whatever you can think of. So obviously that the model is limited in the ability to model that. And also where you have the most obvious problem artifact that we have in imaging is partial voluming, when you have contributions for several components in the brain. And eventually the measured tensor averages the contribution from all the cellular compartments in the tissue. And when you look at tissue microstructure, this is a microstructural probe, you have many compartments that you averages all of them. And at the end, DTI doesn't model restricted diffusion, which we're all aware and saw in the last talks, do exist in the brain. So taking the all complexity, and when you go to the brain, this is not a phantom, this is not just even the nerve, there is a mess. There is a mess of information, many cells, many cell types, a lot of structures, a lot of things, and just taking all of them into one single tensor could be too simplistic. So about 15 years ago, together with Peter Basser, we took a lot of information that was gathered beforehand together with Yoram, and we constructed a model that tried to combine the two things. So the model was called the CHARM, the composite hindered and restricted diffusion model of diffusion. We just so it's a biophysical model, so it tries to model how the tissue looks like and then see what will be the diffusion properties of each of the components. So it just disintegrates the tissue. So if this is a white matter tissue in the brain, it disintegrates it to the components. So you have intracellular, you have extracellular matrix, you have intraaxonal space, and the idea behind this model, we try to make it as simplistic as possible, that part of the diffusion that happened in the intracellular and extracellular spaces will be hindered, and part of it will be restricted, which should happen within the axons. And the model, as we said, we try to do it as simplistic as possible, just contain two phrases. One of them is for hindered diffusion, which dominates at low Q values or low B values, have a Gaussian displacement distribution and can be modeled by a diffusion tensor, while the other part of the equation will be the restricted component that dominates at high Q values, has a non-Gaussian displacement distribution, and can be modeled, at least within white matter, as restricted diffusion in cylinders. And we do have solutions for that. For the tensor, we have the diffusion tensor imaging framework, and for restricted within cylinders, there are several frameworks. We choose to work with the Callahan framework about diffusion within impermeable cylinders. In terms of acquisition, because you need to monitor these two types of diffusion processes, you need to measure, again, to be rotational invariant in many directions and in many B values. So this is a multi-shell, multi-B value, multi-directional approach, which lengthens the acquisition time, which was quite a challenge in the beginning of the experiment. It took 30 minutes, 40 minutes to get a data set. I think today a CHARM data set is done within 6 minutes, 7 minutes, it's done. But the amount of information you can extract from it is quite impressive. So you can get since we are looking at the restricted component, we can look what happens in the axon and within the axons themselves. And then the orientation definition or the ODF becomes much sharper than what we are using in diffusion tensor imaging. And then we can extract information about much better. We can model the effects of crossing fibers. We can also get information about the diffusivity of different parts. For example, you can look at the diffusivity of the hindered part, only removing the information from the axons and look at process that happens there if that's of something of interest. And also, as was shown before, we can extract the restricted fraction, which assumed to be related to the cylindrical orientation, and we can get information on that. So axonal density or neurite density or whatever. If we go back to one of the examples about the dynamic brain, we do know from histology that the things that happens in the brain, at least following the experience, not at the time of the experience, one of the sources for the mean diffusivity reduction is cell swelling of the astrocyte, cell swelling of the astrocyte, and formation of new processes. So the entire tissue becomes dense in a way, and that happens in the human brain when we saw the reduction in the mean diffusivity. Using CHARMT, we were able to see that the changes in CHARMT in the restricted volume fraction happens at the same, it's co-localized almost perfectly with the areas where the mean diffusivity was reduced, but the amount of restricted diffusion increases dramatically. So you have more restricted diffusion in the tissue that causes reduction in the mean diffusivity, obviously related to the increase in tissue density and the formation of new processes of the glial cells, which happens within minutes following cognitive experience. So increase in neurite density, increase in packing of the tissue can be reflected in the CHARMT and give us better biological interpretation to what we see. We saw in the example of the dynamic brain that there are changes not only in the gray matter but also in the white matter, and those are more even harder to give interpretation to, because FA is not a well-defined parameter. It's fractional anisotropy, and you cannot know for which of the biological components in the tissue it relates to. And we do know that FA can go down in neurodegeneration, but it can be lower due to some other processes in the brain. If you have larger axons or non-organized white matter, it can go up, and it doesn't have a good biological interpretation. And it's not only that, it's also very heterogenic. So we have axons that varies in sizes and density in the brain, and this is something that needs to be modeled. So together with Peter, again, after we came up with CHARMT, we extended the model in order to monitor also axon diameter distribution. So the idea behind CHARMT, without going into the mathematics of it, is that you look at the diffusion process in the brain at different diffusion times. So you increase the diffusion time, and in each time, larger axons become restricted. So for example, if you do the experiment at short diffusion time, only small axons will be restricted. And as you increase the diffusion time, larger axons become restricted. And by doing a multi-B value, multi-diffusion time, multi-direction experiment, you can extract the axon diameter, as was shown by several people before. So that was first implemented on the Corpus Callosum. We did validation on nerve sample and phantoms, etc. But the first in vivo demonstration was done by Daniel Barazani, and we tried to see if we can get the information on the Corpus Callosum, just because the Corpus Callosum is a very well-defined structure, is a large structure in the brain, and we do know how the axon diameter distribution within the structure looks like. So this was the first example that we have done, and what you can see here, that different location in the Corpus Callosum, you get different axon diameters. In the genu part of the Corpus Callosum, narrow distribution. In the splenium part of the Corpus Callosum, again, narrow distribution. But in the middle in the body, you get a broad distribution, which is well known from histology. So that could replicate in vivo information that was so far we were able to see by histological means. And the fact is that once we can measure the axon diameter and DTI together, we can see that the relations between them are very problematic. So there is some kind of relation between FA and I mean axon diameter, but this is not a strong relation, meaning that there are many factors affecting FA that are not related to axon diameter, not related to axons at all, as Christian showed yesterday. Taking that into better understanding white matter plasticity, So these examples that I showed you before, when you try to look at what happens when the mean diffusivity of air or reduces or air or FA increases within the white matter, so we do know from histology that there is increase in myelin content in the tissue, but how that affects axon diameter. So this kind of experiment showed us that there is changes within the axon diameter distribution. So when you have a cognitive experience, something happens to the axon, something happens to the axons, which we couldn't understand directly, and I'm not going to go into that at the moment, but a lot of discussions, especially with Heidi Johansenberg from axons, led to the assumption that maybe there is a production of new myelin in the tissue, especially in non-myelinated axon, that increases the amount of conduction velocity. So AxCaliber was first implemented in phantoms and then in animal models, but again, the challenge is to bring it to the human brain. And it is indeed a challenge in order to move from animal models and preclinical scanner to do AxCaliber in a human brain for many reasons. So to move from a mice to clinical scanner, and especially in such impressive brains, could be a very big challenge, especially because of the gradient technology, which was a long time ago wasn't that strong as we have today. But without getting into technical details, there are many limitations to doing AxCaliber in human, but you can get some kind of information, knowing the limitation, but there's some kind of information you can get. And indeed, we can follow within the human brain also the structure of the corpus callosum when you have narrow distribution at the ends of the corpus callosum and border at the body. We do know, and you can take this factor, these measurements of axon diameter as another in vivo microstructural neuromarker or biomarker to look at pathological conditions. And if we assume that in multiple sclerosis, FA changes represent something that happened to the tissue, we might be able to say something more if we look at axon diameter. And in this study that we're doing in the lab, it is not published yet, we do see that in multiple sclerosis subjects, in the normal appearing white matter, it appears that the axon diameter is smaller than in controlled subjects, meaning that there's something abnormally developmentally in the white matter of this subject that might increase the vulnerability of the tissue and later on causes the disease. But axon diameter is a good marker. It's a good marker of the tissue just because it can relate us to functional features of the tissue, because it is well known for many years that the axon diameter helps the axon conduct the velocity faster. So the larger the axon is, the faster the conduction velocity will be. And this was something that we wanted to demonstrate in the human brain. So in that study, we combined electrophysiology measurements of ERPs with measurements of axon diameter. And in that study, we used the ERP in order to look at interhemispheric transmission time, IHTT. So how much time it takes to move the information from one hemisphere to the other hemisphere. So we used the visual task and a tactile task to stimulate one visual field, then it transfers through the corpus callosum to the other side. And by measuring the ERPs, you can calculate the difference of time, which will give us the IHTT. Doing a sort of combined analysis, taking fiber tracks and the length of the tracks and the IHTT, you can measure the conduction velocity, the axonal conduction velocity, and try to correlate it with the axon diameter, as we measure it with AxCaliber, combined with all the limitations that are in clinical scanner. But you can find a good correlation in several domains, and you can see that you actually find that the ratio is the literature ratio that we know about conduction velocity per micron of micrometer of axon diameter. Well, the next steps of it is to move into 3 dimensions, because AxCaliber was a framework that was developed in 1 dimension, but our brain, lucky for us, is 3-dimensional with many fiber systems going in plenty of fibers with a lot of strange curvatures. And the idea was to expand it. So in the last few years, we tried to come up with a framework that will enable us to extract the full axon diameter distribution for any fiber system in the brain. So the framework itself, it's quite complicated, but eventually you can get this kind of map that just maps the probability of small axons versus larger axons within different white matter fascicles. So it was validated on the well-known fiber system in the Corpus Callosum, etc. But eventually we can get maps like that. So if you look, for example, of fraction of small axons in the SLF, the segmentation of the SLF into the 3 segments or segments becomes evident, because you can see that the axon diameter properties in each of the segments, it's different. Or for example, if we look at the IFOF, is a fiber system that relates many cognitive domains, but you can see that you can segment the IFOF into several domains based on its axonal diameter properties along the tracks, as well as other domains. For example, you can see the sensory and motor fibers have higher, larger axon diameter than other fibers going. So obviously, using axon diameter can give us better input for connectome analysis, so we can try to look at that. So with that, I will summarize. So diffusion imaging is great. Diffusion tensor imaging is great. It made huge advances in neuroscience, but we do need, it's high time that we'll move beyond the tensor, just because we can get better biological interpretation. And people in neurobiology, whenever I give this talk, they said, so what does it mean? Mean diffusivity, what does it mean in biology? What does FA mean in biology? And we do need to give them better answer than tissue density and packing of axons. So we know now that biophysical model of diffusion in neural tissue, like the charm and other that people will speak later on, can give better characterization and interpretation of the diffusion MRI signal. And the higher sensitivity and specificity that we get from this model that go beyond the tensor can enhance our understanding on these processes of the brain, the dynamicity of the brain, the connectomics of the brain, and neurodegenerative conditions. With that, I would thank the people that work with me over the years, and collaborators, and students, and friends, and thank you.
Thank you very much. I think we have time for a couple of questions.
I almost know what you are going to ask.
I wanted to say, Yaniv, you know exactly the question I'm going to ask. So can you answer this question?
So for the benefit of the audience, please ask the
well,
question.
so what's your idea of why you have a certain discrepancy between histological diameters and the ones that you're measuring? I mean, it's there, right? Let's acknowledge
There is a discrepancy, obviously. So
Yes.
Can I answer it? Yeah, please go ahead.
First, I would say exchange, but it's not really the case. Histology itself. If you think about the way how the neural tissue is actually prepared, you quite often, and in regular standard histopathology, you shrink not only the tissue, but not equally well. And that's the major problem. How you can, how we can fix the tissue to preserve the geometry.
Does it shrink by a factor of 3? 5?
That is exchange. So, yeah. So, as a matter of fact, it might. It really.
Can I listen to it? Well, just to say something, the first posters that I presented at the ISMRM, I stood back to back with Greg Stanich, that was in Sydney, and 20 years later, we are still back to back in this meeting. So, yes, the measurement of axon diameter depends on many technical factors of the scanner. You need very high gradient strength. You need a lot of stuff to do very short TE, whatever that you cannot meet at the moment. We are getting better. We are getting better with the technology, but we cannot meet. So obviously that causes some kind of bias in the measured axon diameter, and the bias that is that we need to better understand it, because our ability to infer the exact size of large axon is much better than the ones of smaller axon, that's for sure. But just bear in mind, as Greg said, histology is not the best parameter to look at axon diameter, because when you fixate the tissue with formalin, it affects differently, just because formalin gets in touch with the membrane. So if you have more membrane in larger axon, they will have more formalin fixating, and that will shrink them again. So there are some biases in any measures that you have in neuroscience. Once you know the biases, all of them, and you're able to put them in your interpretation of the data, that's okay. There is no method in neuroscience. Neuroscience is not an exact sciences. That's it. We need to be aware of it.
I think we have one last question.
Yeah, it's just a comment, an additional comment. I think behind the technical challenges, which you are completely right with that, I think we should not forget that the model is a diffusion-driven model, but the T2s are different between the compartments, and this is not taken into account. So depending on the TE, depending on the magnetic field, the balance will change.
Definitely, definitely. So a model is a good model, but it's only a model, and obviously it can't model everything. So you can incorporate T2 if that's the case, but eventually we'll have just too many parameters to fit. But yeah, you're all right. So, but the model gives you something. If you believe the model gives you something significant, then go ahead. If not, throw it away.
Thank you very much. Thank you very much, Yaniv.